In this video I'm going to explain what whole cell voltage clamp is. I assume that you already know what voltage clamp is. I cover that in a separate video. So, whole cell voltage clamp comes from the realm of patch clamping. So you might already be aware that with patch clamping what you do is you have a cell, such as a neuron, and the cell has its ion channels, and what you do with patch clamp is you make a glass electrode, like a very fine glass pipette, a bit like this. So this is in cross section, and then you fill it with some sort of salt solution, and then you bring that towards your cell and you push the electrode up against the cell like this. So these are the two walls of the glass electrode. This is glass. So it's like a really fine glass pipette, and this is the salt. And what you do is you take this and you compare that pretty much with what you record outside the cell, and that gives you your signal. And if you've got just one ion channel underneath, you will see the ion channel opening and closing like that. So this is what we call cell attached. Now, you can now do another thing. What you can do is you can break this membrane underneath, and you can usually do that by applying a bit of suction or a pulse of voltage. And what that does, it breaks the membrane underneath. So you've now got a low resistance access to the inside of the cell. And this is what we call whole cell. The advantages of this is that this resistance here is very low, so we've got good quality access to the inside of the cell. So now what you can do, you can apply a voltage clamp here, and that gives you a good quality voltage clamp of the neuron. The old way of doing it would be to have two what we call sharp electrodes to pierce the inside of the cell like that, and another electrode here, and this one would be used to measure the voltage, and this would be used to inject the current to achieve the voltage clamp. The problem with that is, first of all, you need two electrodes, and getting one electrode into a cell is hard enough. Shall I just write that again? Two electrodes, and getting one into a cell is hard enough. Getting two is even harder. And two, it's a high resistance, which is good here, but not so good here, because you're trying to send in a lot of current through a very narrow gap, if you like. It's very high resistance. So for all sorts of technical reasons, that's not a good idea. This is a low resistance, so we can get a good quality voltage clamp. If we wanted to, we could actually switch from voltage clamp to what we call current clamp, which is the same thing. We adjust the voltage so that there's no current, and that enables us to record things like action potentials and EPSPs and IPSPs.